Fusion, the international science radio show.
Hello,
Hi,
how are you?
good, thanks. How are you?
Lovely to meet you.
Yeah, nice to meet you. Hi, I'm Marsilea Booth, and I'm a senior scientist here at Nutromics.
Looking very quickly at the paper, the impression I've got is that you've been developing a patch that can detect concentrations of drugs in human tissue. Is that roughly correct?
That's nailed it. Yeah. So we're making a medical device, and exactly as you say, we're firstly looking at the concentrations of a drug in particular called vancomycin, as it is in humans. So this was the first time that it had ever been tested in humans.
And vancomycin is a particular antibiotic?
It is, yeah, it's an aminoglycoside antibiotic. It's very commonly given in the US. In Australia, it is also given, but not as commonly as in the US.
And I think, is that because it can affect more serious illnesses and they don't want resistance?
Yeah, so it's actually given to to make sure that resistant bacteria, infections of resistant bacteria, so MRSA in particular, is why they give vancomycin. I think in Australia they don't have as much indication of it, and it can just be like a choice of the country of what the recommendation is. In the US, they give it a lot.
Right. So it's important to be able to work out how much of the antibiotic is in someone's bloodstream and in the tissues.
Yeah, so vancomycin in particular, it's known to have nephrotoxic complications if it's given too high. So it can induce acute kidney injury. And that's one of the challenges of it. You need to make sure that you give enough so that you're able to get rid of the infection that may cause problems in the body, but you also can't give too much or otherwise it can cause damage to the kidneys. So what we were really trying to do is give clinicians an idea of how much is in the body when they need that information. And normally the way that it's looked at is just through a blood draw. So they're looking at taking a blood sample and then measuring how much vancomycin is in there. We're actually measuring just below the skin in the interstitial fluid. And that's interesting for two reasons. One, that it's non-invasive, so you're not having to go as deep in, you're not having to use as big needles. And the other advantage, perhaps, is that we're getting information about what's happening in the tissue, which is often where the infection is. So you're actually measuring the concentration of the drug where you want to be measuring it. But all of this is very new. So plasma is what's more commonly looked at.
Right. So you've got a very different sort of sensor involved here.
Yes, we use electrochemical aptamer-based sensors. These are based on some technology that came out of UCSB with Professor Kevin Plaxco. And they're really interesting. They sort of were inspired by nature. Instead of just giving a response, the way that they actually measure how much vancomycin is there is by changing their shape. So it's a selected DNA strand. You might think about antibodies that you hear about quite commonly, and they're able to find a specific target. This is kind of similar, except it's a synthetic, like a man-made DNA sequence, which is able to change its shape depending on whether or not your target is there. So when vancomycin is there, it has a certain shape, but when vancomycin is gone, it's also able to release it. So in that way, we can measure both rising and also falling concentrations in the body.
Right. So it actually changes its shape, and does that change the electrical signal that you can detect from it?
Yeah. So I probably have oversimplified it by saying it changes its shape. There's quite a few different things that can happen in there. And actually, we don't 100% know. There's still literature coming out about what's happening to that DNA strand. But whatever the change that happens when the target's there, the main influence that we're measuring is a redox reporter that's at the end of that DNA strand. So this redox reporter is methylene blue. And how close it is to the electrode surface will give us a change in signal. So if the vancomycin's there, it may bring that DNA strand end close to the surface. That redox reporter is close to the surface, and you get a change in signal, and that's what we're using to measure.
Right, right. And of course, so you've got electrons coming from the end, and you're
Yeah,
detecting that with the electronics, and that tells you
that's
how
right.
much
Yeah.
of the drug is present or not.
Yeah, so that methylene blue, that redox reporter, we're actually using the electrons from that as it gets oxidized and reduced. And the way that we're doing that is having a potentiostat, a way to apply voltage and measure current in the patch itself. And that's something else that was new in the work that we're doing. Quite commonly, you're having to use these bulky commercial potentiostats that take up a lot of space, have a lot of wires. We're, for our product, developing everything small, similar to what you'd see in like a continuous glucose monitor, where everything is contained within that single patch. So that was really exciting to have it all together.
That is quite amazing. So you've got this tiny little patch with everything in the one place. And how do you get just under the skin to that fluid level that you need to measure?
Yeah, so we do have small needles on there. They go in about 3 millimeters deep, which some people understandably aren't keen on needles at all. And we have just 4 of them. And it was one of the things that we were really interested as well. Within the team, there are people who their work has a lot to do with usability and also patient experience. And so we really tried to capture that as part of our study as well. What do the participants feel when they have it inserted? How does it feel during it? Does it hinder their mobility? And then lastly, as it's removed, are there any lingering effects, or how, in their opinion, is there pain? And thankfully, everyone rated it very low, which is great.
Oh, fantastic. So let me think about this. So how did the ideas for this, to make it this small and to go for this level and this type of thing come? Because I know I had a very quick look, and you've been working on sensors and things for a long time.
Yeah, me personally, or the company you're interested in? I'm happy to discuss that.
Well, whichever you have a story to tell me about.
Well, let's go with the company first. So
yes.
I sort of touched on it before, but part of the reason why we wanted to go just below the skin was to make it minimally invasive. So it's not going to be going into the blood. It's not going to have to go too deep in it, and it won't hinder the person. And part of that, for vancomycin, you're doing it in an ICU setting, but the patch that we're making is really a platform device. I mentioned before about a DNA strand that's being used to capture your target. If you change that DNA strand out, you can look for a different target instead. So it's really a generalizable platform. And part of the reason why Peter Vrains and Hitesh Mehta started Nutromics was sort of as preventative medicine. At the moment, quite a lot of what we're doing in hospitals is reactive. We're waiting till we see the symptoms. We're waiting to give any of that treatment. But they were really interested in preventing deaths by looking at proactive preventative medicine. And so eventually the idea would be maybe these patches could measure a target that could be used for consumers, for everyone around, to make sure that they're not getting sick and finding out before they're sick.
Right. And so that's the company. And what about you?
So I've always been really interested in sensors. I think from when I was sort of young, I always wanted to find out why things happened, what's going on, more information. I guess that's the scientist in me. And then when I was an undergrad, I started to hear about these biosensors and how they can really give you the information that you want. You can get more information, and then also you can get it when you need it. So I did my PhD working on DNA sensors, so sort of similar, but instead of trying to target a molecule or a compound, I was targeting just other DNA strands. And that was in academia, then moved to a small stint in industry, also looking at biosensors. Moved across to the UK, and there I started working on enzyme sensors. So those would be a lot more common that you guys would hear about for the continuous glucose monitors. And those can work really well. The challenge with those is that you're using enzymes on your sensor surface. And that means that it needs to be a naturally occurring substance. And it means that it really limits how many things you can target. You can basically get enzymes for glucose and lactate and some other ketones. But beyond that, you can't really do it to look at synthetic drugs or a range of compounds. And so that's where the beauty of this technology comes in. Because you're making the DNA yourself, you're actually able to target a number of different compounds in a really broad range.
So you can expand this to all sorts of different drugs, not just to the vancomycin.
Yeah, so in the literature, there are quite a few groups who are working on EAB sensors. In Kevin Plaxco's lab alone, I think they've got maybe 24 different targets that they've looked at. And it ranges from being drugs to also some metabolites. And there are a number of different compounds that they've made sensors for with different potential applications. It's one of the beauties and also one of the challenges with this kind of sensing, because the beauty is that there are such a broad range of targets that you can go for. The challenge is narrowing down on which one is going to have the biggest impact, and which one is going to be able to help people, and then as a company, which one makes strategic sense.
Mm. And now that you've completed this round of clinical trials, the silly question that we always ask is how long would it take for this to get to the market?
No, it's not a silly question. I think it's a fair question. So this paper that we've published is on our first in human. So that's a healthy participants. And it's really exciting to see the results, because there aren't many literature examples of what's happening in ISF. So this is the first step towards like showing that it can work in a healthy cohort. So the next step is really, well, what does this look like in a setting where we want to use it in an ICU setting? And so those clinical studies, we've started some of those in Australia now. And we're hoping for product maybe at the end of next year, but that's our sort of planned pathway, that we will get these ICU study data and then analyze that. And yeah, exciting to see.
Fantastic. Support diffusion by buying from the Diffusion Science Etsy store. Catch people's eyes like flies. I have Hawaiian shirts, t-shirts, pullovers and mugs with many different Venus flytrap designs based on my photographs. Check out the shirts and other Venus flytrap designs at diffusionscience.etsy.com. I'm Ian Woolf. So what are you targeting next?
Yeah, so we're starting off with vancomycin, but one of the things that vancomycin can be used to treat is suspected sepsis. And we think that there's going to be some really interesting work in trying to identify from the literature and from talking to clinicians what kind of targets would be interesting for trying to see if we can identify whether sepsis is present. Part of the trouble with sepsis is that some of the indications can be very broad, and also the timing of when you need that information is really important. And so if we're able to sort of find targets and make sensors for those targets that could identify the presence of sepsis, then that would be fantastic.
And so sepsis is when the immune system's overactive. Like, can you just explain?
Yes, yeah. I'm not going to be giving a very good explanation of that, sorry, because my background's more on the sensor side, but that's also my understanding of it, that sepsis is when the body's inflammation takes a sort of overreactive response to an infection, and then can cause, really serious damage.
Well, that's right. So my understanding is it can cause damage, and if it's left unchecked, it can kill you.
Yeah, that's correct. Yeah.
So you would be able to, well, you will be looking at different molecules that the body releases when it's going through sepsis to be able to work out that that's what's going on and diagnose it more.
Yeah, so there's a panel of compounds that are normally tested for the orders given by clinicians when it's a suspected sepsis case. And so some of those might be things like lactate or CRP, C-reactive protein. And so those might be things that we kind of look to next after we've got our first product.
Amazing. And very important.
Yes, yeah.
So you will be able to look at biologicals as well as drugs. Well, would you be able to look for things like viruses or bacteria as well?
Yeah, that's an interesting idea. So as I mentioned before, it really depends on the aptamer sequence that you use. There's a special process that we use to identify what that sequence should be, and that's called CLEX. And theoretically, you could do it for many different targets. In reality, and practically, it can be a challenge. So something so big like a bacteria may be too challenging for aptamers. Although, again, in literature, people have definitely tried it. And viruses, I think, again, the size of it would be a hindrance. But yeah, again, look to literature.
So we don't see a lot of patch electronics in everyday medicine. Do you think that's something that's going to change a lot?
I think it's moving more and more towards wearables. Both like you see everyone walking around with Apple Watches or with other watches that are collecting data. I think as data becomes more and more important, as people are able to control more of their own health as well, I think it's going to become more and more important and more visible everywhere.
Because one of the things that has come up is like you've got people living with diabetes are now having those little implants in their arm to monitor their blood glucose and insulin. Would it be a less invasive way if there was a patch for that could detect what was going on?
Yeah, so I would call it a fairly similar patch, except that current CGMs go quite deep compared to these needles. But definitely, if we are able to, there are people working on trying to make needles that go less deep than current CGMs to be able to measure glucose. So I think something that would be able to be less invasive would be adopted by the community, provided it can give accurate results. And also last the distance, because those ones are sort of up to 2 weeks now.
Yes.
And so that's something else. So in our paper, we look at only a 24-hour period of our EAB sensors, our electrochemical aptamer-based sensors, the patches on people. But current CGMs are up at the 2-week mark. So again, that's something that we looking for other products we might have to look into more.
And the sort of molecules you're working with for your sensors, is that nanoscale or is that a bigger sort of scale? Is that micro?
So the sensors, sorry, you talk about the needles and the sensors or the targets that we're looking for?
I guess the way you're looking for the targets.
Yeah. So everything's happening really close to the surface on the sort of nanometer scale because you've got those DNA strands. They're quite short strands. So those are really not very far from the surface itself. What we're relying on is really the diffusion within the body. So the interstitial fluid, which is where we're measuring, it's sort of outside of the cells. And what we're really looking for is the free drug moving from the blood into the interstitial fluid and then diffusing to the sensor surface, measuring it. Again, one of the beauties of it is as that concentration near the surface depletes, as the body excretes the vancomycin, that the aptamers and cells actually let go, dissociate the target and probe dissociate, and we're able to measure that decreasing concentration as well. A few years ago, I won the fellowship to be able to work at Imperial College. And that was a great experience. I worked across 2 different labs that were very cross-disciplinary. So one of them was a very census-focused lab, and the other one is the lab of Dame Professor Molly Stevens. And she works with so many different areas from biomaterials to sensors to just across a huge range. And so through that, I really was able to work on my communication, working on projects with very different areas, because often that can be a challenge. Just the language, one word in different groups can mean completely different things. So, and then also I think project management, it really helped with, because you are collaborating with lots of people, which is great. And also prioritization and time challenge. So it was a great experience to learn there.
Fantastic. I thought I'd seen something about a 2025 one, but maybe that was somebody else. Was there like a and no? Obviously, no.
Not me, I think.
Obviously I got the wrong information.
Well, no. I'm trying to think if there is anything, because I've been at Nutromics for nearly 5 years now, 4 and a bit years. I've been mostly like in industry. We don't apply as much to those sort of external ones, which is kind of unfortunate. But I'm very excited that we were able to get this publication, because again, in industry, it's not always common to be able to publish your work. So,
and this is a Nature journal, isn't it?
Yes, Nature Biotechnology. So that was really exciting as well, having it out there. A few people have asked me, well, like, why were you publishing? You're a company. And I think it's really important because it gives visibility to the scientific community outside of Nutromics to see what we're doing and how we're doing it. Secondly, this is new information. As I mentioned before, measuring in the ISF is challenging. There's not much literature out there that does it. Maybe microdialysis, but that has its own challenges. So this information is new and exciting for maybe people who do pharmacokinetic modeling. And then also, it's like a nice gathering, because we mentioned before about how there's the electronics and also the DNA, and then also the sensors themselves. Like, it's a big group of different people along with our academic collaborators. So being able to publish it as a group is really nice.
Yes. And how did you find the switch from academia to corporate work?
Yeah, there's pros and cons to both. I am not one of those people who's kind of had my career organized out from day one. I'm very much a see what the opportunities are and say yes to them. So I went from doing my PhD academia to industry, back to academia to do a fellowship, and then now back to industry. I like the idea with industry that there are multiple of us all working towards a shared goal. We're very strong at Nutromics on what our vision is and what we're trying to do with our work. I can also focus on the science, and then someone else's job to sort of maybe look for funding or to, all of those sides of the work. Whereas in academia, I think you sometimes have to wear so many hats to be able to even get the funding to be able to do the science. It can be a challenge. And saying that, there are some nice pros as well, where you're able to have more control over what it is that you do, and also be able to publish, be able to collaborate and go to conferences. I've been really lucky in my experience so far at Nutromics, where they are encouraging of that sort of personal development and also academic collaboration.
And if any listeners wanted to focus their studies in your field, what would you recommend to them?
Yeah, so I started out very focused in chemistry, and then as I've progressed my career, kind of moved more and more towards bioengineering and that cross collaboration, so the multidisciplinary, interdisciplinary work. I think saying yes to opportunities, recognizing opportunities, is a really important one. And then also, I think it really helped me to have that strong fundamental understanding. So really learning that chemistry deep, and then how it can be practically applied before kind of making the move to application of it, I think has really helped me as well.
Fantastic. And let me think. Can I ask you about some other projects the company has to keep them happy?
Yeah. So specifically, like what else the company is doing with things?
Yes,
or...
what other devices that they have? Or are you
we're very
project?
So we're working on the patch, and that's our aim. So that's what we're doing for our product. We're still fairly new. Yeah, so about 2019 is when we started this technology, using this technology. The company was founded in 2017, but 2019 is when we started working on this technology. And sometimes I feel like it's shifted a lot, like we've done so much, and then sometimes I see how much more we have to go. It's exciting, but if we look from where we started, which was very much in the academic, like what they published in academia, where it is individual sensors that you're making by hand, each of them individually. If you are going to put it into, for example, preclinical, you would have to use a needle to put the wire in place, the wire sensor in place, use these bulky electronics. And I think it's been a huge leap to now have this all in a single device, where we can sort of semi-automate some of the manufacturing, and also keep it all in that single patch.
Because it sounds like you've had an interesting career, and that this is an interesting turn for you. But
it's actually quite a nice sort of comeback, because when I was doing my PhD, I saw some of Dr. Kevin Plaxco's papers, and I remember thinking, like, this is really exciting science. Like, how are they being able to measure in animals and people, and sorry, not in people at that point, only in animals at that point, and on the bench. Because I'd experienced with my sensors that I'd worked with so many problems when you're putting it in a real biological fluid. You've got so many, like a soup mixture of all these different things in blood that affect your sensor response. Even if it works really nicely on the bench and buffer, when you put it in real systems, you can have a huge challenge. I remember going to a conference and hearing from one of Kevin's postdocs and asking them afterwards, well, how are you not having problems? Like, how are you overcoming drift? And I think the answer was something like, it works. It just works. So they have like a KDM, kinetic differential measurement, which is what they use to compensate for drift. And it really does work well. I guess in hindsight, having something that is inspired by nature, being made from DNA, which is in nature, we shouldn't be so surprised that it works well in the body.
So what's drift?
So drift is anything on your sensor that doesn't come from target. So any response that you see from your sensor that doesn't come from the vancomycin there. So it's a problem that a lot of sensors have, and one of the ways that you can compensate for it is mathematically, and that's the way that our sensor system works.
So it's sort of like a type of noise.
Yeah, so it's like a type of noise, but it can be just also over time. So it's not necessarily something that can happen for a short time. It's, for example, temperature could affect your sensor signal, or if the sensor breaks down, that would be called drift. If some other, I think those are the sort of main examples of drift.
Right. And so your work is mainly on the molecule end, or are you working on every aspect of the device?
So I started off in the lab working on the sensors themselves, trying to make sure that we could make the sensors last as long as they possibly could, and give as high a response as they could. As my career's progressed, and once we started to work with the clinical team, I became more of a liaison between the clinical and the sensors team, and then now bringing in the patch and the engineering. I'm sort of a go-between for those three, but still with sensors at my heart of that foundational background.
So multidisciplinary.
Yeah, I think I would say that I refuse to not be included. I'm always interested in all of it and want to hear about everything.
I think that's a really important role just in itself, isn't it?
Well, I like to think so. I think sometimes it can help with the conversation. So, for example, one thing that I really like to do is I like to draw graphics. And if I can, in a graph or in a schematic, capture a scientific concept that will help across the different teams, I think that's helpful. So I like to do that.
Well, it's because you're bridging different ways of looking at things as well as different domains of knowledge. It's almost like a translator role, isn't it?
Sometimes it definitely feels like that. Yeah. As I said before, like a single word, I think we all got caught up on the word sample because in software engineering, it can mean one thing. In chemistry, it means quite a different thing. So being able to, make sure that everyone is speaking the same, understanding the same thing by the term is really important.
I think so. So we've talked about the next steps, and we've talked about how you got here. I'm trying to think if there's any other questions I should ask while I have the chance.
What's your background in science?
My background, I originally had a degree in applied physics, and I worked for a very small amount of time in a solar observatory. And then since then, I've been doing a weekly science and tech show on community radio and podcast.
Yeah, I listened to some of them. Like I feel like your role must be similar because you are also having to translate from a very detailed science, someone who speaks just generally detailed science to the broader community, which is also really important.
Yeah, that's the type of role there for sure.
Yeah.
Well, all right. So, Marsilea, if there's not anything else I've missed asking you that you'd like to talk about, is there anything?
I think I've covered. Yeah, I'm happy with the questions.
Right.
And
well then, Marsilea, thank you very much.
No worries. Yeah.
Study science, because you will find in the study of science a richer, more rewarding life.